Hello. So now the next two lectures is going to be about signal processing. So the first one is going to be about one-dimensional signals, and the second one about images and how to analyze them. And so for next Tuesday for the tutorial, if you can pick an image or a signal or one-dimensional signal that you like, and then think about how to analyze it, and then we can all discuss it of different approaches to analyzing them. So that's for next Tuesday, and that's actually so the second part of the homework is that. So there are lots of different signals. So for example, last week Stuart talked about ChIP-Seq. So this is one type of signal. So you have so it's a one-dimensional signal. You have an axis. In this case, it's genomic coordinates, and then you have a measurement of that has peaks and valleys and signal and noise. And what we're going to talk about today is, for a few different cases, how we can, by looking at signals, figure out what is the signal and what is noise part, and then separate the two. That's sort of the main aspect. So both this lecture and next lecture. Another signal, a little bit older data, is from ChIP-Seq. So here it's a time course. So we have again on the x-axis genomic coordinates. So this is chromosome 16 now in yeast. And then we have time taken different time points through the cell cycle. And what we see here, and what we're looking at, is a replication fork associate protein called GINS. So GINS, in the beginning, we don't measure anything but noise pretty much before in G1. But then when the cells enter into S phase, we start seeing that GINS binds to origins of replication. And some origins, the binding starts earlier and others later. And then what we see is that GINS start accumulating at the origins and then start moving away from it. And finally, it's there's not much GINS left at the origin, but between origins. So what we see is that GINS accumulates at origins and then moves out as replication happens. So what we see here is that this is definitely the noise here in the beginning. Then we have the signal here in the middle overlaid onto the noise. And then at the end here we see these spikes, which is another kind of signal. We see some of them here also, but they're clearer down here. And then again, we only measure noise afterwards. So this is just another signal that we want to be able to look at. And then we've already looked at this in the proteomics section. The mass spectrometry is another one-dimensional signal that we want to be able to analyze. So again, here we have peaks, and if we zoom in, we have lots of details. So each peptide has an isotope distribution. And then we can zoom in even further, and we see that we have nicely separated isotope peaks. And in this case, a strong signal, so there's not much noise. But if you look at another region here, we see that even in this region where there isn't much signal, first, if we zoom in, we see that there is a lot of signal. So here again, we see the isotope distributions of the peptides. And we see that the curve is a little bit more jittery. So here we have more noise. But what we also see is that there's pretty much a peak at every mass. which is a different kind of noise. So pretty much it means that this is a very complex mixture. So there's a background of many, many different peptides at very low intensity. So we need to deal with this kind of noise in a different way than when we look at noise over here in the chip experiment before it goes into S phase. So these are just a few examples. So the basics of signal processing is we can decompose signals into different components. And so one basic component is a sine wave. And you all know the sine wave. It starts at 0 when the angle here is 0. And then so it's defined as if we have a circle with a radius c, and we define A, B, and C, the sinus of the angle alpha is A divided by C. So what it means is that when we move this line through the circle, we will get an oscillation. And we can define the amplitude and the wavelength here. And then we have the cosine, which is just the sign slightly shifted. And it's instead of A divided by C, it's defined as A divided by B. So now if we take two sine waves and add them together, now they have two different frequencies, and two different amplitudes. So these are the two sine waves. We add them together, we get something that looks like a little bit a noisy curve. So now, in cases when we have simple oscillations, a simple sine curve, we can see that it's periodic and it's nice. If we start adding up many different frequencies, it's not so easy to see what the different what frequencies it can be decomposed into. But there is a method called Fourier transform where you can go from the linear space into frequency space. And here, the definition here involves integration and complex numbers, and it's pretty much you take the original signal, f of x, and you integrate from 0 to infinity with this factor in front of it, and then you get the decomposition. But the main thing is that you go from the real space to frequency space, and it's very easy to implement it in different programming languages. They have defined functions to do this. So we're just going to look at what the effect is here. So now we made this composite curve adding up two frequencies with two different amplitudes. So if we do the Fourier transform of this, we get back two peaks at the correct frequencies with the correct ratio between them. And so the one thing, when we do this, plot this, we plot the absolute value of this. So remember, so we had here, since we're going from real space to a complex space. So we have both a real and an imaginary part. So we have to get the frequency distribution and the relative ratios here. We need to take the absolute value of the Fourier signal. So then the other thing is the phase that we also have. And that's because we need to define where each of these start. So whether it starts here as a sine wave at 0 or whether it's shifted away. So that's why we need the complex space. So now, so the nice thing is here, even if we have quite a lot of different frequencies, we're going to, in frequency space, be able to see them individually and see what their contribution is. And the other thing is that, of course, we can go from real space to frequency space and then back again. So if we take this and do the inverse Fourier transform, we get back the same signal as we had before. And so the reason to then do this transformation is, of course, we can then have better control over how we filter our signal. So in this case, if we take the whole frequency space and do the inverse Fourier transform, we get back our signal. But if we just take one part, the low frequency part, we get back the low frequency component. And if we take the high frequency part, we get back the high frequency component. And this is something that we're going to use later on to filter our signals. So sometimes we want to remove the low frequencies, sometimes we want to remove the high frequencies, and so on. And we can sometimes remove the extremely low frequencies and the extremely high ones and just keep the ones in the middle. And that depends on the application and what we want to do with that. So but what we often want to look for is a peak. We want to find peaks. So in ChIP-Seq, you saw that we wanted to find where there are peaks, and we want to, in mass spectrometry, we also want to find peaks. And to describe a peak, there are several things that we can use to describe it. So one is, of course, what's its height or what's its area, and exactly where it's located. So meaning that either where the peak is at its apex, or where the average, so the centroid of the peak is. Another thing we're interested in is how wide the peak is. And that we can, for example, define as the full width at half maximum as the peak width. So, because that's the nice thing is that's an easy thing to measure. You see, just take the half height and measure how wide it is. And then we can have other measures like mean variance, skewness, kurtosis, and so on. So these are all things that we might be interested in knowing about the peak. So if we look at first the mean and the variance, it's quite straightforward to define what that is. If we have f of x is our peak, and we normalize it so that if we sum over all the points of the peak, it's equal to 1, then we can easily define the mean as the weighted average of the points within the peak, and the variance is also easily defined. So now if we want to start looking at different types of peaks, so one is Gaussian, which is a normal distribution. We can easily generate that in Python, and we can also take the Fourier transform of it in this case. So here we have then we see that for the Gaussian it's dominated by low frequencies. There's quite a lot of intensity here, and then there is not really very high frequencies, which we also can see it's quite a low frequency peak, as can be seen here. So that's the basic peak, and we're going to work with this peak quite a lot throughout the lecture. We can then relate the full width and half maximum at half maximum to the standard deviation. The area is related to the height and the standard deviation and so on, and it's symmetrical, so there's no skewness and also no kurtosis. That's the way. So it's a nicely behaved peak. Now there are other peaks, for example, we can define a peak with a longer tail, which is this is called the Lorentzian, and it has other names also. So the functional form is given here, and you see that it has a much longer tail, and actually the mean and the variance is not defined for this one, but we can get the median, and of course we can get the full width at half maximum. That's related to, again, and we can calculate the area. But one important thing to do later on when we're trying to look at our data is what do our peaks look like? Are they nicely behaved Gaussian shapes, or do they have other problematic features like very long tails, or whether they're maybe skewed, like in this case? This is an example of a skewed peak, where you see that the tail in one direction is much longer than in the other one. And again, here it's always showing the Fourier transform of the peak, and then we see that this one has a higher frequency component when we compare it to the Gaussian. So now, since we always have both the signal and noise, we're going to look at normal noise. So this is just simply Gaussian noise that we have generated with Python's random number generator. So we see that we have this random noise, and if we look at it, we plot it as a histogram, this deviation from zero. We see that it looks nicely distributed as a Gaussian. And if we do plot it on a normal probability plot, it will follow the straight line, so we can really know that this is nicely Gaussian. So this is just a check of what that. And if we then take the Fourier transform here, what we see is that this is pretty much all the different frequencies are. So that's why it's also called white noise, because we have just contributions of all different frequencies. So we always prefer if our noise is normally distributed, because then we can then we have methods that can handle that much better than otherwise. So if it's not, then we should try to transform it, and that's sometimes possible and sometimes very easy. So the one thing that you've come across already is the log-normal noise, which is the case for a lot of genomics and proteomics. So here you see that, again, we generate this random noise, but here it has very large outliers. And we again see that the distribution is far from normal. And then it has a lot of all the different frequencies, but then it has also a peak at very low frequencies. So, but the nice thing with the log normal, when our noise is log normal, is that we can just simply take the log to get the normal. And that's why we, in microarray analysis and proteomics, we always work with the log of the signal. Then we could have skewed noise, which could make, and if we don't recognize this and think that our noise is normal, then we can often think that we're detecting signal, but it's really that the noise is skewed. So in this case, we see, you can probably even see that this noise looks different than it seems to have more structure and more peaks, but it's just that there are more outliers because it has this skewed form. And that's the very tricky thing to try to characterize and see if your noise is normal or if it's not. If you have a lot of signal, you can often do it, but it's not straightforward in general. Okay, so now we can add these together. So we had a Gaussian peak. We looked at that it had low frequencies when we moved into frequency space. We had our normal noise, which is just a flat distribution of frequencies. And then if we add them together, we get a noisy peak, and we get all the frequencies overlaid onto our peak that corresponds to the Gaussian signal. So that, and that's what we now are going to try to recover the original signal when we have. What we can do, like we did for the sine waves, is if we go to in frequency space, we can slice it and remove high frequencies. So if we start with removing all the frequencies higher than, in this case, 30, then we see that we go from this very noisy to the black curve here, and the red shows the original Gaussian. But so the black curve, it looks much nicer, much less noise, and our signal is much clearer. So then we can go down and remove everything above 10, and now we have a very nice smooth signal, a little bit of noise ringing left over here, but not too bad. But then if we go a little bit further and cut probably a little bit too much, we cut into this what corresponds to Gaussian, then we get widening and higher and several low frequency signals that we add on top. So that's pretty much. So here we probably, if we cut at 30, in this case we didn't smooth enough, but if we cut down here, we smoothed it too much. And probably around, for this case, around 10 would be the ideal, where we can reconstruct the peak quite well. So another concept that we use often in signal processing is convolution. The way it works is described pretty well in Wikipedia here, and that this figure is from there. So if we have a signal f, and we convolute it with g, which is, so this is a square signal, and here we have a triangular signal. So what we do is we take g and move it over f. So we start out here when they just touch. Oh yeah, so the other thing we do is we invert the direction of g. So we flip it. And so instead of being triangle in this direction, it's now a triangle in that direction. So then we start to move it in over f, and then just calculate the area under the combined signal. So what we get is a signal that's f convoluted with g. And so the nice thing here is that if we do this in Fourier space, so if we first transform both f and g into Fourier space, we can just do a pointwise product when we do it in frequency space. So that's, and then we can transform it back. So that's a very, that's why it's defined this way. So what we can do with this convolution is we can do smoothing. So instead of moving to Fourier space and cutting, we can do convolution to some kind of shape. So either just a box, triangle, or in this case a Gaussian, or even more, there are other shapes also that people have used. So this is the one example. So we have our peak with noise, and then we do different smoothing with this box smoother. So again, if we just do a little bit of smoothing, we still get quite a bit of noise. If we do too much smoothing, we get a wider distribution. So now, if you look in Fourier space, here we can't really see the difference between the two, and actually it's pretty difficult. Even here in this case, you probably might see there is a gray. No, it's not. Sorry about that. So before, when we did the smoothing by cutting in Fourier space, now we're just changing, weighting the different frequency components and not doing such abrupt cutting. So these are just some examples from mass spectrometry then. So here we have an isotope distribution from a peptide, and then we add some width to that peak and noise. And then if we then run a smoothing filter over it, we can recover the peaks, and not perfectly, but quite well, especially the two first ones. So here is another example with another heavier peptide. Again, we can clean up the signal quite nicely by just applying a little bit of smoothing. Even in cases when we have really a lot of noise, for example here, in this case we can hardly see the signal. But by applying a smoothing filter, we can at least recover the first two peaks of the isotope distribution, even though they're very wide and probably not that, but it's still much better defined than what we can see from the noise before the smoothing. And another case where we barely can see the peaks in the original signal, but then we can lift them out with the smoothing. So another thing, another filter that came from photography, it's unsharp masking or adaptive background correction, which can be used to separate peaks that are overlapping. So an example here would be that we have this peptide, but the peaks are wide, so we can barely see that there are three peaks, but they're just small shoulders on top of the first peak. But if we then do a combination of smoothing and in this case actually only adaptive background correction, we see that we can actually lift out the three peaks. But as you see, they're not very well defined. The first one is close to 0, but this should be at 1 and that should be at 2. So we might be able to lift out the peaks, but we also introduce a distortion. And this is just another example of that, where we can separate the peaks, but we distort their positions. And then the same thing with noise. We have the now noisy peak and do adaptive background correction and smoothing to get at least the first few peaks in isotope distribution recovered. So this is something that one can has to sort of look at the signal, try different settings, and it should also only be used for really for looking at it, for visual inspection. If you want to quantify the signal, it's often distorts things more than is useful. But to inspect the signal, it can be very useful. So this is one example that we'll come back to next lecture on the where we have a noisy situation. We have 4 peaks. We have one wide peak in the middle that we're not interested in. That's the noise. And these 3 thinner peaks are our signal. So what we do here is we smooth it so that the wide signal gets that the baseline follows the wide signal, but it doesn't go into the small signal. So if we subtract now this red line from here, we get this situation where we have the 3 peaks, but it's very noisy. So after smoothing, we get the 3 peaks, and we can set a nice threshold to detect just those. As you see here, we get around these 3 peaks, there is an overshoot that it goes down to negative when we do this. But this is actually the homework also to run a script and find the parameters where you get this situation that you can remove the large peak, wide peak in the middle, but still keep the thinner, smaller peaks. So now we have a lot of the tools that we need to start detecting features. So now what I want to demonstrate is sort of a general strategy for what to do when you have a signal. So what I'm proposing that one should start with is to characterize both the signal and the noise, and see and that's what we're going to do for this simple case of a noisy situation where we have a few 3 different steps, some bigger than others. And so then we can, when we have characterized and understood what the signal and the noise looks like, then we can make a model of the data, and we can also select, we also need to select the detection method, and then we can do simulations and to select parameter settings for how to set thresholds. So if we characterize the noise first, so we want to see if we're lucky, it's going to turn out to be normally distributed. So first we have to remove the steps, and we can do that by just subtracting a moving average, and that will remove and we only have the noise left in this case. And then we can make a histogram of the noise and see that in a normal probability plot it looks nice and linear, so it's normal noise. So that makes the situation much easier. Now we need to model data, and just by looking at it we see that these are steps. So we can then just introduce a step. So now when we're going to do the simulations, we're going to put the step in the middle at 0. And we can also have different signal-to-noise ratios. So this is signal-to-noise ratio 1, signal-to-noise ratio 2, and so on. Then the last thing we need before we can start defining parameters is the detection method. So anyone would suggest a method for detection that we can use? Okay, so a simpler method would be just to calculate moving average in two windows and take the difference between them. So when we are done, when both windows are on the same side of a step, the difference will be close to zero. And then when they are on different sides of the step, there will be we have a value, and then it again goes down to zero. So we're going to convert the steps into peaks in this way. This just shows what the result of this. So now we've changed a few different things. So first we have different signals of different signal-to-noise ratios, and we also have selected different bin sizes for what to do the averaging over. And so what we see is that if we choose a very large bin size, we can often detect the step in the right place, but the uncertainty is large because we don't have a nicely defined peak. But then, in the case of when the signal-to-noise ratio is pretty high, we can use a smaller bin size, but not when we have just the signal barely above the noise. So that all makes sense. So now we can start doing the simulation. So the first step we're doing here is to just look, where do we detect the signal? Where do we detect the step? If we have a signal-to-noise ratio of 1 for any of the bin sizes, we almost always detect it in the middle. But for lower signal-to-noise ratios, like here 0.05, it's pretty much random. There's a little bit of preference in the middle, but not much. So now the nice thing with the simulations here, now that we've decided on how to model our noise and our peaks, is that we really we can explore the parameter space. So another thing that we're interested in, how often do we get the correct peak? So now we just say that we're going to take the largest peak as the position where we think it is, and how often is that correct? So we have now the correct is the red, and this is then the score, which is the height in this case. So we have if we have a large bin size and signal-to-noise ratio 1, we always succeed. If we have a low signal-to-noise ratio, 0.05 here, and a small bin size, we almost never succeed to find the right peak. And even with a large bin size here in this case, we don't succeed very often. So, but we can then say from these kinds of simulation, we can then tell what kind of signal-to-noise ratios can we at all detect with some certainty. And then the next thing is that how do we set the threshold? So that and what we want to control for is usually the false discovery rate. So how many times do we get the peak position wrong? And so this just shows the simulations looking at the same signal-to-noise ratios and a range of bin sizes, and showing what the false negative rate is, the black. So what happens is if we increase the threshold, the false negative rate goes up always, and the false discovery rate goes down. So what we need to decide is what false discovery rate can we tolerate, and then we can set the threshold that way, and then we also know how many, what our sensitivity is, how many times do we actually succeed to find the right, correct signal. So this, I think, is quite a general strategy of approaching when you have a signal, on how to start thinking about it. And it's not always going to be this easy. This is a very basic example, and often it's much more complicated. But it's at least a start how one can approach the problem. So let's talk a little bit more about peak finding. So in general, if you have a nicely behaved peak, you probably all know that if you take the derivative of the function, then it's going to be 0 at both minima and maxima. And then if you take the second derivative at maxima, that's going to be negative at maxima. So what happens here is that if you take the derivative at a point, this is now positive, then it's at the peak it's 0, and it's then negative. So that what means that the second derivative is negative at maxima is that you go from positive to negative in the derivative space. But real signals, this method is often not so applicable. Maybe in some cases you can do a lot of smoothing first and then look for derivatives, but usually it's not so practical. But we can again then apply our same strategy of we're going to characterize the signal and the noise, make a model of the data, select the detection method, and select parameters using simulations. So first we're going to characterize the noise. And now the easiest way is we can just ignore the peaks. They're there, they're just a few, 3 small peaks, the rest is noise, maybe we do fine. So we can just see, and what we see is that it works pretty well, but we have this quite ugly deviation from the in the normal probability plot that is due to the peaks. It actually turns out that the position is not 0, it's upside down here, but it's 0.04 instead of 0, and the width is 0.26 instead of 0.2. But it's still pretty close. But then what we can do is we can calculate the root mean square deviation. So we select the window and just add up calculate the average within the window and add up the squares of the deviations from this average within the window. And then look for outliers. So we're going to see that there are 2 peaks corresponding in the root mean square deviation, and that corresponds to these 2 peaks. So we're going to remove those and then recalculate. So this is then what we removed, what's in black, and then we're going to redo the calculation on the noise, that's the red. And then we get a much better fit and actually a much better both width and location where the noise should be. So it did matter, even though it didn't look like much, we would have gotten quite a distortion if we wouldn't have removed the peaks. So now to characterize the peaks, we of course can do the opposite. We already know where they are, so we can just inspect them. And they look like this in this case. So they're not very, we don't have that much statistics. They look a bit noisy. It's difficult to say their exact shape. So the default is, of course, if we don't have any evidence to the contrary, we can assume that they're Gaussian, is what we're going to do here, since we don't have any other evidence. So now we can model the data. We had the noise, and we have a model for the peaks. And so we can simply add the signal to the noise and have different signal to noise ratios. And now to the detection method in this case, we can again, yeah.
So what you got these two numbers S over N. What is, how do you estimate S and how do you estimate N?
So I estimate the signal as the height of the peak, and the noise.
You assume you have a single peak, and that's the signal, and then N is what, sorry?
Is the standard deviation of the noise. So that's the, and of course that can be discussed how to define that, but in this case I mean, yeah, so one has to define it some way. So now, in this case, one detection method would be just to, again, have a window and just calculate the moving average within this window size of choice. And then, and that will give us sort of a smoothed version of that, and we will get a peak that we can, a detection method that we can work with. And then we can define that we want to find peaks that are large. So what we're going to not do now, but what one can do in the same way as in the step detection, is to go through and do the simulations and to determine what parameters, what signal to noise ratio we can detect for the peak, and also what at the set signal to noise ratio, what would be our false discovery rate and our false negative rate, and so on. So that we can do in a very similar way as we did for the steps detection. But we're not going to do that now. And so again, after, just to repeat, so the information about the peak that we're interested in, after we've gone through and detected it, is to characterize it, where it's located, what's its height, because often or height or area, because often what we have is that we can use peaks for quantitation, that if we have a large peak, it means that whatever gives rise to that peak, there's a larger amount of it. And often we try to work with different measurements within the linear range, so that if one peak is double the other, it should mean that there's double amount in that case of the what we're measuring. So just quickly to summarize what I've talked about today is we have a very useful tool, the Fourier transform, to move between real space and frequency space, and to both to characterize the signal, but also to do different kinds of filtering. Then we want to characterize our signals and to really understand what is signal, and then decompose it in what is signal and what is noise. And we want to, when we have characterized it and understood both the noise and the signal, then we can model it and do simulations to understand, compare different detection methods, but also determine what parameter settings we should use for the detection. We then talked a little about filters. Mainly, we talked about low-pass filters, so we removed the high-frequency component to get to remove noise. We talked about a few different detection methods, mainly based on moving averages, which are probably the simplest things that we can use. We talked about convolution, And actually related to convolution is something called correlation, which is we can use that to measure similarities between signals. And an extension of that is autocorrelation, where you correlate the signal with itself to look for periodicities that might be obscured by noise. And we actually didn't talk about how to do coincidence measurements, but yes, so that's the summary of today's lecture. And so next time we're going to add on one more dimension and talk about how we can apply some of these ideas to images. Thank you. And also look at the homework and bring your favorite signal next Tuesday, and then we'll discuss that.